Okay, I'm gonna slowly start. Well, more people are joining. My name is Thibaut Goldsborough. Today I'm going to be introducing InstanSeg. I'm a PhD student Peter Bankhead is my supervisor, who's the man behind QuPath. So today I'm going to be presenting InstanSeg, which is our new embedding-based nucleus and cell segmentation algorithm. I'm going to introduce how the method works, but also how you can use it yourself. I'm going to focus mostly on Python, and hopefully we can train our own model in real time by the end of this workshop. I'll also show you how to use InstanSeg in QuPath if you don't want to touch Python. That's also fine. What is cell segmentation? I suppose if you hear it that you all know briefly what it is, but the idea, we want to differentiate background from foreground. We also want to separate touching objects. It's a really difficult task. There's a whole range of different images at different resolutions. You can have fluorescence images with a black background, and with many, many different channels, or you can have standard bright field images, RGB. So it's a very challenging problem, and we built InstanSeg to try and address or at least help the segmentation of cells. So I'm going to start with the Python install just because it could take a minute or two. If you want to follow in real time, you can. I'm going to try and install everything from scratch, so without assuming that you have nothing on your computer. So all you need is conda and git. If you don't know what conda or git is, that's fine. Just hold back for a few minutes and I'll come back to the presentation. So I'm going to click presenter mode. So I'm in my terminal and I'm going to go. So if you go to the InstanSeg GitHub page, you can go ahead and copy the git clone. So I'm going to, in my documents project, I'm going to go git clone and then the link. This could take a few seconds. Then I'm going to create a new environment. I'm going to start from scratch. So I'm going to go conda create new. I'm going to call it InstanSeg demo, but go ahead and call it InstanSeg if you want. And I want Python, say 3.11 in this environment. Just take a second. Then I'm going to activate the environment. And then I'm gonna so I've cloned the repository so I can walk into InstanSeg, and you can now set up this as a pip install. So you can go pip install e.full. So this is just for detail, this is if you want a full InstanSeg install. Otherwise, we're on PyPy, and you can go pip install InstanSeg torch, and that's fine as well. Just saw the question in the chat. Oh, can you zoom in a little bit? Sorry, I don't think I can. Yeah, I don't know how to do that. Really sorry. I'm going to go back to the PowerPoint so you can see the commands I do. But I'm doing pip install e.full. And that will install InstanSeg. So that will take a minute because I'm installing Torch in the background. So I'm going to jump back to the presentation. How does InstanSeg actually work? So InstanSeg relies on a unit backbone, which is very lightweight. It's convenient if you're running InstanSeg across full slide images. And the idea is that it's embedding based. So the unit gives an embedding to every pixel. And the idea is that embeddings that belong to the same object are close together in embedding space. And embeddings that belong to opposite objects are far apart. Now, InstanSeg doesn't use a clustering algorithm to cluster these algorithms, to cluster these embeddings. Instead, we use a neural network. And this neural network basically takes two embeddings and says, yes or no, these two embeddings correspond to the same object. The advantage of this is that we essentially use a neural network for our post-processing. So there's no watershed, there's no flow tracking, and there's no iterative algorithms. So that means the entire pipeline is essentially just matrix operations, and that has huge performance implications. So I'm not going to bore you too much, but we benchmark InstanSeg training from scratch on a range of public data sets. So hopefully I'll show you how to train your own model on TNBC in just a few minutes. Again, just showing that InstanSeg performs well when you train it from scratch on these public data sets. Now, because InstanSeg is fully vectorized, it means the post-processing is extremely fast. In fact, the whole pipeline is at least 60% faster than other cell segmentation methods that we know of. It's about 10 times faster than cell-based. And it's not necessarily because our model is smaller. Actually, our model is similar in size to the cell-based model. It's because the post-processing is highly optimized, highly vectorized. Now, it's not only speed the advantage of having a fully vectorized pipeline, we can also package it in this object called a torch script. So this is an object that lives in the C++ world, and we don't really have to worry about this. But the huge advantage is that it can live outside of Python, which means we can run InstanSeg directly in software. So for example, here I'm running InstanSeg in QuPath, which is a Java-based software. So unlike other segmentation methods, InstanSeg is doesn't necessarily have to live in the Python world. So I'm going to go back to hopefully this is finished. And I'm just, so here you can check in your environment that you've got PyTorch with CUDA installed. We can do it, so I can go Python, import torch. And then I can check that CUDA is, so CUDA again is a language for running on a GPU on a graphics card. Is going to accelerate InstanSeg massively because everything's vector-based. So I can go is available. And hopefully it'll say true. If it doesn't, you're going to have to uninstall PyTorch and then follow the official instructions on the PyTorch website. Great. So now I'm going to open, I use VS Code. So I'm going to open the folder that we've just downloaded. And inside, so inside the repository, so yes, I trust the authors, there's a notebooks folder. So in here, you've got a segmentation minimal notebook. And this, for example, so I can start running, so I can call InstanSeg. I've got a bit too many. Oops. Sorry. Okay, I can't find the repository that I just made, but I've got a backup one. So I'm just going to use a backup repository. Sorry about that. And then, so in a single, so here I'm just downloading an example image. So nothing to worry about. I'm just going online. I'm taking this great image from PerkinElmer, which is shared with a CC BY license. And then if you, so you can import InstanSeg as a class, and then you can automatically download. So we have two public models. We have a brightfield model and a fluorescence nuclei and cell model. So if you run this command, it will go and fetch it automatically from GitHub and download it locally on your computer. So I can go ahead and, so if I want to call InstanSeg, there's this eval function, and then it's going to save the predictions to disk. Now, often you want a bit more granularity, a bit more control over what's going on. So you can do this in steps. So what's happening in the background, InstanSeg is reading the image as an MPI array, and it needs the pixel size of the image. So it goes and reads that from the metadata of your image. You then send that information to the forward function, so this eval image, and it gives you a labeled output. So again, this is a NumPy array. So I can run that. And then I can make a display and then show the display. So if you want to know what's happening behind, I've got this function called show images, and you can label output. So this is what label output looks like. But if you want to have a nicer display, you can use this display function. So here I ran InstanSeg in a single pass, which means the entire image goes to the unit. If you have a bigger image, so this is the fluorescence image that we just downloaded. Once images, so this again was in a single pass. So InstanSeg is really GPU efficient. So I have a relatively small GPU, and I can do this in one go. But if you want to chop it up into small tiles, if you have a really big image, then you can go ahead and call eval medium image, which works just the same as eval small image, except in the background is going to chop it up in small tiles. And here you can give parameters like tile size equals 512, batch size equals 3, and I can just run that again. So it always takes a second or two to warm itself up, and then usually accelerates. Yeah, and so here I was predicting in green and red nuclei and cells. Here I specify that I only want the cells. So it only gives me the cells, and it gives random colors, but the labeled output won't be random colors. It's just a display. If your image is too big to fit in RAM, we have this eval whole slide image, which I'm not going to run here, but essentially it'll read the image in small tiles from disk. It won't read the entire image into disk, and then it'll instead of returning like a NumPy array, which would be way bigger than RAM, then actually stores it in memory as a ZAR array. So it works similarly, but it's very much an experimental feature. So I'm going to go and jump straight ahead. And what if you want to train your own model? So these we have pre-trained for you, and they're publicly available under an Apache 2.0 license. But say you have a data set which has a non-commercial license, or a data set which is only in your institute, then you can go ahead and train your own model. So for this, you need to download a public data set. So we made a notebook here explaining how to do that. So you have to run the first cell. So this sets a few background variables. And then, for example, you can run any of these dataset cells that you want in any order. It doesn't matter. So I've got TNBC, which is a nice one. Cell pose, there's some manual steps to do because you can't download this automatically. But then, yeah, you can plug and play, download any dataset that you want in any order. So I'm going to just download TNBC because I'm doing this live. So this is what InstanSeg wants. It wants a pair with an image and then a labeled array. This is what InstanSeg wants for training. So you'll notice that this doesn't allow overlapping objects. So once you've downloaded the image, you can scroll all the way down to somewhere called save all the datasets. And so here you can run this command, and it'll save your dataset to this object here. So it'll appear over here under InstanSeg datasets, then you've got this object, instanseg.pth, segmentation dataset.pth. So if you want to train your own model, you go back to the command line. So you go to cd instanseg script, and then I'm going to go and say -train.py. I'm going to call the name of the dataset. So we said it was called segmentation dataset.pth, and then I'm going to specify the name of the specific dataset that I want to train on. So This object can contain as many datasets as you want. So in our case, it only contains TNBC, but it can contain any dataset. So here I'm going to say I want to train on TNBC and TNBC only. For this demo, I'm just going to say set E, so that's the number of epochs. I'm going to set this to just 5 epochs because we wouldn't have the time otherwise. And then another parameter is the pixel size. So this is the pixel size that I want to run InstanSeg at. So during training and during testing. So for example, if I said 0.5, that would be quite efficient for inference across a whole slide image. But if you want more granularity, you can set it to, for example, 0.25. And the unit is microns per pixel, which is fairly standard. So I can click enter. And it'll think about it. And I've got a CUDA out of memory. That's because I need to restart this environment. And I'm going to try again. Again, I've got a small GPU. Great. So now it's training. Here it's saying it's saving results to this path here. And the name of the model is called My First InstanSeg by default. But of course, you can change that from the command line. So this is going to take a while to train, and I'm going to go back to the presentation. Yeah, so InstanSeg can be run directly in QuPath as well. And if I have time, I can show you at the end how to do that. But InstanSeg, if you don't like QuPath and you don't like Python, InstanSeg is shared in the bioimage.io format, which means you can plug it in directly into Fiji with the deep image J plugin, and without any specific post-processing, you can run InstanSeg in Fiji. But this won't do tiling for you. It will work well for small images. It might not work for a whole slide image. So I'm now going to move on to a slightly different part of the presentation, where I'm going to show why InstanSeg is uniquely suited for whole slide images. Sorry, for multiplex images. So that's images that don't necessarily have 3 channels. They can have many, many more channels. So here's an example here from the CP-DMI dataset from Alanik et al. And there's many, many different channels. And it's not completely obvious if you run a deep learning model, which channels do you give to the deep learning model? So for example, Mesmer or Silpos or other algorithms, you're going to have to select 1, 2, maybe 3 of these channels, or you can combine them craftily to make different pseudo images like here on the left. And then you're going to give these pseudo images to your deep learning model. But this is a manual process, which is very much error prone. So we developed a new deep learning algorithm called ChannelNet. And essentially it's a unit-based algorithm that will take any number of input channels. We've tested it all the way to 150 channels. And it's invariant to the ordering of the channels. So you can have DAPI in the first position or the last position, it doesn't matter. And output of this model is going to give a fixed channel representation. So as a simple example, I can show an RGB image. So 3 channels output. And this is going to be the input of the main InstanSeg network. So the idea is that we can then train this model by only optimizing cell segmentation in multiplex images. So we have no control over what this intermediate representation is going to be. Only that is going to be informative about the boundaries of nuclei and the boundaries of cells. So what does this look like in practice? So again, this is the same great data set from CP-DMI. And we take these 8-channel images. You can see the ordering is not necessarily preserved. This is not a nuclear channel, for example, at the bottom in blue. But we, for each of these combinations of markers, we can make a fixed RGB representation of these markers. And then we can give this to InstanSeg to predict both nuclei and cells simultaneously. And this means we can use InstanSeg on multiplexed images without having to retrain the model for novel biomarker panels, which is really exciting. So if you look at what these intermediate representations look like, they indeed have the nuclei appear all in one channel, in here in orange, red, and then the cell membranes appear in dark blue. So it confirms a hypothesis that yes, it can work. These channels do contain information. Sorry, this intermediate representation does contain information about the boundary of cells, which is really exciting. So we can look at an extra experiment. What happens as you increase the number of channels at test time? So here, what happens if you give it only a single channel? Star, this is a DAPI channel, so it does contain information. The accuracy is quite good, but then as you add channels as input, then the accuracy goes up. Now, take this with a pinch of salt. It doesn't necessarily mean that the more channels you have, the better segmentation you're going to have. For example, all of these 14 channels don't necessarily contain only noise. So I'd recommend not running InstanSeg on just noisy channels. So I can go back to so if you go back to the command lines, InstanSeg's been training in the background. And we hit an F1 score of 0.33, which is not terrible, but it's not very, very high either. So we can have a look at what this looks like. So projects, InstanSeg, and then it saves into this folder called models, and then my first InstanSeg. So inside here, you have the weights of the model, but you also have the outputs of each epoch. So I can show it. This was the first epoch, the second epoch, the third epoch. So red usually just means empty. So the top left is the input image. The second one on the top is the ground truth, and then the third one is the prediction from InstanSeg. Again, so I'm training this in real time. You can't expect to train a deep learning model from scratch and perfectly just in a few minutes. But if you train this over just a few hours, then you can reach state-of-the-art segmentation accuracy. So any questions in the chat? So, okay, so Ian asks, if your objects during model training have different pixel size scales, is this problematic for InstanSeg training? For example, microscope images taken with different objectives. So that's a good question, and it's when you load the data set, so I'm going to go back to my load datasets notebook, you actually specify, so InstanSeg reads the pixel size of the image. So InstanSeg will store in the metadata what pixel size this image was imaged at, and then we'll rescale this image so that it matches the pixel size that you are requesting during training. If you train it on your own data set, so here I give like a very rough skeleton. So if your images look like this, are in this format, then you can run this function. You can run this cell, and it will make a data set for you. And you have to specify the pixel size here of that image, or you can read it from the image metadata, whichever you prefer. So hopefully you know the pixel size of your images. If you don't know the pixel size of your image, so I'm talking, for example, of the cell pose dataset where they deleted all the metadata from all the images, then you can set the label area to like a fixed number of pixels. So you can say the median label in my ground truth is going to be 300 pixels. And to see how to do that, you can look at the load cell pose code, because we do this in the background inside this function. But ideally, you haven't deleted the metadata from your image. There's another question. Is it better to fine-tune InstanSeg or to train a unit from scratch while using InstanSeg for generating training data? So that's also a really good question, and probably an open research question. I always train my InstanSegs from scratch just because they take so little time to train, and I've got access to a compute cluster. There's also the risk that if you're retraining an InstanSeg is going to forget the original data sets that it was trained on. So I always train InstanSeg with all the data sets, including a custom data set that you may have. But this is an open question of an open research question. You'd have to try both and see which one works best. But I would train from scratch every time. But you don't have to. In the Python repository, it allows you to restart training from a checkpoint without any issues. So I'm going to look how far this is going. So now our F1 score has now gone up to 0.42, which is now fairly reasonable. So again, just in a few minutes, this is what it looks like. So I think it's ready to export. So there's another notebook here called export model. So I'm going to run this cell. Cross my fingers. Perfect. And then we can call this function called export bioimage.io, and this will take your training your InstanSeg. So this is the model that's training in the background. I haven't cut it yet. And I can go ahead and export it to bioimage.io format. So before I do that, I'm going to cut training because my computer's going to run out of memory. But I can go ahead and call this. Yeah, so there's a question here. Is the IOU calculation done at 0.5? No, it's done so we calculate the IOU between 0.5 and 0.9, and then we take the average of all the IOUs. So that means an IOU of 0.5, sorry, an F1 score of 0.5 is already a very, very good F1 score. So, I mean, not like incredible, not like benchmark breaking, but it's already like a decent F1 score. So here, again, I just trained it for about, 10 minutes, and it predicts more or less the Sáenz is making lots of mistakes, but here we've got a bioimage.io models, and then we've got our model, my first InstanSeg. So you can use this exactly as all the other InstanSeg models that you've downloaded. You can put it on the bioimage.io archive, but I'm going to show you now how you could run this model, for example, in software in QuPath across whole slide images. So I'm going to launch QuPath. Okay, this is QuPath. And I'm going to go to extensions, InstanSeg. Before I do this, I'm going to open a project. RTK demo. So here I've got different images. So I'm going to open the Luca1 image, which is the same image I showed in Python from Perkin Elmer, CCBY, and well, actually, I'm going to open a brightfield image and show. So this is another image, H.E. Hamatsu from Cambridge. So I can, it's a TMA array. It's huge, but I can. So before I do this, I'm going to restart all of my. I'm going to release memory because my computer's struggling. And you can go ahead and select a region, go to extension, InstanSeg, run InstanSeg. And then here you can select, so you can point it to your bioimage.io folder. which we just created earlier. And then here is going to appear my first InstanSeg. So this is the model that didn't exist a few minutes ago. We literally made this, we started training 20 minutes ago. And so it's already packaged and I can click run. So QuPath will read the pixel size of your image, and then it also knows the pixel size to run InstanSeg app. This is stored in the metadata of your bioimage IO model. And then here we go. We have already predicted a few hundred objects. In QuPath, you can decide whether you want the objects to be full or not. Now it's making plenty of mistakes. Again, it was trained in a few minutes live, so it's not going to be the most incredible model. But we also provide some pre-trained models which are publicly available, and they have a nice license. So so I've got a cloud. This means it's coming from my repository, from our repository on GitHub. So brightfield nuclei. It's gonna download the model and run it. And then here's going to make hopefully a lot less mistakes. So if you're wondering what datasets it was trained on, you can click on the little eye and this will open the README. So you can see what datasets it was trained on. You can see that they're all CC BY or Apache. That means there's it's open to they're properly open source datasets. So if you want to see, if you're more interested from the fluorescence world, so we can switch back to the Luca image. So this is an 8-channel image, and it was trained on. We can use this other model called fluorescence nuclei and cells. This model, we can click on the eye. It was trained on CPDMI. This is amazing data set. So if you use this model, please cite this article as well. And then I can select a region. So here I'm doing Control-Shift-A, that selects the whole image. And then it's asking me what channels I want to give. So here I can select every single channel. And then the outputs. So do I want nuclei or do I want cells? Or do I want both? So I'm just going to leave everything default. It's running on the GPU, which means it'll be fast. It'll be relatively fast. Okay, so this is the output. It could be a little bit confusing. So I can select, for example, only the cells. And you can see there are some little issues. For example, here we've got a cell inside a cell, or almost inside a cell. So this is because QuPath doesn't allow for nested. It doesn't allow for multinucleated cells, and that means if we find 2 nuclei inside one cell, we're just going to create a new cell that corresponds to that nucleus. This is to make all downstream use in software much easier. So if you don't want this behavior, then you can only predict cells, and then you won't have this this strange artifact. So I'm going to try and it's still open. So I'm going to click run again. And then we don't have that strange artifact anymore because now we only predicted cells. Now you can also click measurements, and QuPath will make all sorts of measurements about the fluorescence intensity of all these cells in the background, and then you can use this and export it. So I saw there was a question, can you export the QuPath segmentation that InstanSeg generates as labels to import into another software? So yes, of course you can. I don't use QuPath that much, but file export, oh, there you go. So you go File, Export Objects as GeoJSON, and this will export a compressed version of all the labels. So instead of storing like a TIFF image, which could be way bigger than your system memory, it will only store the outlines of the object in a GeoJSON format. So yes, the answer is yes. Of course, you can run this in QuPath in Python as well, and then Python will give you the option to save either as GeoJSON or to save just as a regular TIFF image or any NumPy or ZAR array that you want. So because InstanSeg is channel invariant, we can play around and see what happens as we unclick these. So for example, I can go to view brightness contrast. I can remove, so we can only, so this is DAPI, and we can add, for example, CK. So say we only have a 2-channel image, then I can right click and then select from visible, and this will select only the 2 channels which are visible. And then I can likewise click on run. This time it's making all the measurements in the background. And this, yeah, so this is the output. And you can play around with this as much as you want. You can switch off channels that are that contain a lot of noise, for example. Now, recurring a question that we often get is how does InstanSeg doesn't necessarily work well with cultured cells, doesn't necessarily always work. So our public fluorescence nuclei and cell model was only trained on a single data set that contained lots of different types of images, but they were all tissue. So they were all tissue sections. So here, this is not a tissue section. So our fluorescence model may or may not work. I can click on image in QuPath, and then it will tell me that the pixel size here was lost somewhere. So QuPath can't read the pixel size of this image. This image is from the CellPose dataset, so they removed all metadata from all the images. So we either have to find what microscope this image was taken and then reintroduce the pixel size, or we can, for example, I can select a cell briefly, say this is roughly the size of a cell, click on pixel size, and then a good area is roughly 100 microns, but this is not, yeah, this is not necessarily very rigorous. It's much better to read the pixel size of the image, but this suggests the pixel size would be roughly 0.2 microns, which sounds right. So now I can select the whole image, go back to my InstanSeg extension, and so I can run this model if I want. It will, again, it was trained only on tissue images, so the output may or may not be very good. It's not terrible. But if you want a better model, you can retrain your own model on, for example, the CellPose dataset or any other cell culture dataset. So I've done this. So for example, we made this cultured cell model. So we can't release this model yet because it was trained on the CellPose dataset, which has a slightly ambiguous license. But you can retrain this yourself. We explain how to do it in the notebook. So if I click on run, then the output is a little bit better. It actually finds the outlines of cells. But again, we can't, at the moment, we can't really share this model publicly because it's a bit ambiguous on the license. But you can do it yourself, and you will download the data from the CellPose host. And so you can then decide under what terms you want to use this model for your own interest. So yeah, I think this is the end of my presentation, so I can answer questions if you need. I know I went very, very quickly. If you want, you can go to our GitHub page, which is InstanSeg. So you can go to GitHub InstanSeg, and that will, yeah. So you can go to GitHub InstanSeg, and that'll explain how to redo everything that I just did at your own pace later on if you want. Oh, so really interesting question in the chat. So is InstanSeg trainable using sparse annotations, or do you need a fully annotated data set? So it's actually a good question, and I'm looking into this at the moment. So the short answer is yes. Actually, so the way InstanSeg works, I haven't gone into details here, but actually we take, by default, we take a subset of the training in an image. Say you have 100 objects. InstanSeg during training will take a subset. It'll take, for example, 50 of these objects for training. This is to cap memory and also to make things to make training smoother. But it also has a huge advantage that you don't necessarily need to annotate every single cell in a crop. The caveat to this is that, so again, I don't really have time to describe the full method behind how InstanSeg works, but essentially there's two components to the loss function, one of which requires every single object to be labeled, and the other one doesn't. So if you have a sparse annotation, it's important that you label what is background and annotated, and what is foreground. So what I mean by that is that you can't just annotate 10 cells and say all the rest is not annotated. InstanSeg needs to know which pixels are background. So you need to annotate, yes, so we have this, so the mask is already implemented, but you need to tell InstanSeg what is the mask. So the way, the default we decided to have is that so if you go back to the original inside low datasets, so this is the input. Say you have a region which is not annotated. You can set the pixels to -1. So here all the pixels 0 is background, but annotated background. -1 means not annotated, which means it could be foreground, it could be background, you just don't know. But make sure that you include some background in your annotation. Don't just annotate the objects. Otherwise, InstanSeg will have no idea. It'll probably overswing. However, I said this, we haven't actually tested this, and we don't, yeah, we haven't tested this thoroughly. So we haven't worked on any data sets which are sparse. Yeah, the option is there, but it might need a bit of experimentation to see how to optimize this. But yeah. Any other questions? Oh, I have a do you find yourself training models more often locally or passing the work to an HPC cluster? So personally, I do this on a cluster, just because I don't want my computer to, I want to have access to my computer when I work. So I can train as many InstanSeg models as I want in parallel on a cluster. And this is great for experimentation and for developing the method. So InstanSeg is fully compatible with training on a cluster. For example, you can shut off all of the printing to terminal. You can go to scripts, and then so the train apply has all of these parameters that you can play around with to make it work on a cluster, to increase the number of workers, to tweak the loss function weights, etc. So you can go to train.py, and it's all documented there. Any other questions? Okay. Well, thank you so much for attending. I'm going to stay around for another few minutes if a question comes up. But thank you. Yeah, thank you for attending. So there's a question from Nina. Sorry if I missed this. Is there a notebook to show us how to train InstanSeg using all the data that it's trained on and custom data? So it's not quite a notebook. So there's a notebook called Load Datasets, and that will show you how to download datasets that we use during training. Then training is usually done on the command line, and I do this because I don't like to train in a notebook because I use a compute cluster, so I do this from the command line. So it's documented in our README page on GitHub. So if you want to train your own model, you can. So it's what I just did. Oops, sorry. You can specify what dataset you want to train on, but if you say have multiple datasets, then you just put a comma here after TNBC 2018, and you put a comma, and then you put the second dataset, for example, CellPose, or another, it's a list, so as many objects as you want, as many datasets as you want, and then your own custom datasets. And then it'll train them all. So by default, it'll if you have, say, 1,000 images in the first dataset and then 10 images in the second dataset, there's going to be like a big dataset imbalance, and it'll just treat. It won't rescale them at all. But if you read our training configurations, there's a parameter you can set so that both datasets are called equally. But these are details. There's another question from Clément. Maybe you told this, but can you deal with overlapping cells the way Stardust does, for instance? So at the moment, no. InstanSeg doesn't allow for overlapping objects. So there's a couple of reasons for that. The first is the vast majority of training datasets, training and testing, are flattened. So there's no overlapping objects. So actually, it would be a bit ambiguous to calculate, for example, an F1 score, if you're predicting overlapping objects, but the ground truth is flat. I know this is a bit of a hand-wavy answer. So if I go back to my presentation here, so InstanSeg predicts objects in separate planes. So a bit like a one-hot encoding, but it's actually done sparsely, so we're not storing all of the zeros because that would require way too much memory. So in theory, yes, we could have overlapping objects. In practice, if we want InstanSeg to work on all platforms, so in Python, for example, in QuPath and Fiji, with minimal post-processing, it's really convenient to output a flattened object. Now, if you went in and hacked this very last layer where we basically do an argmax, and you remove this argmax, then yeah, you could have overlapping objects. But then that would probably greatly limit how you export your model in software. But something we should look into, definitely. At the moment, what's slowing us down is that the data sets are all flat, so it's not going to increase the F1 score or the accuracy by having overlapping objects. The other consideration is that there's not all that much. There's not that many tools in software, for example, in QuPath, that enable you to handle overlapping objects. Like, for example, how do you it's not obvious how you phenotype two overlapping cells. If one cell expresses, I don't know, some biomarker, and then it's overlapping with another cell, how do you know that whether the biomarker corresponds to one cell or both cells? This is not obvious, and it doesn't exist really yet in software. So there's not a huge advantage to have overlaps at the moment in software, at least. Of course, you could write your own scripts. Yeah, this is my best attempt at hand waving your question away. Sorry, but no, InstanSeg doesn't yet allow for overlapping objects. Yes, the video should be uploaded, so not by me, but by the hosts, the I2K hosts. And I think it's going on YouTube. Okay, if there's no other questions, I'm gonna go ahead and end the meeting. So thank you so much for attending. If you have any future questions, you can ask on the ImageSC forum, where we're active. You can also raise an issue on GitHub if it's a specific bug that you're running into. Thank you so much for attending.